Mark is a physical scientist in the R&D department of the US EPA. Among his research interests is the use of HRM to investigate the fate and transport of PFAS and other xenobiotic compounds in biological and environmental test samples. Some of his work has been published in peer-reviewed journals, and we are very pleased to have Mark speak on this today in a talk entitled The Discovery of Novel PFAS in Environmental Samples Using High-Resolution Mass Spectrometry.
Hey, Andrew, I can't share until the other speaker stops sharing. It's giving me that error.
I just stopped sharing.
Excellent.
You can take it from here.
Guessing it was you. Okay. Can you confirm, Andrew, that you can see my screen?
I can, but it's on presentation mode. Here it is.
Appreciate
Perfect.
it.
Thanks,
All right,
Mark.
thanks for the opportunity to present. And I'm going to switch gears a whole bunch because I looked through the speaker list and so peptides and proteins and metabolomics. I'm going to tell you about using high-resolution mass spectrometry to discover novel polyfluoroalkyl compounds in the environment. And hopefully you know what these are. And if you don't, I could give a half-an-hour talk just on that. But compounds like PFAS and PFOA are likely to be regulated soon in drinking water with MCL values and reference doses. So I'll give a little bit of intro into defining a couple of terms. Many of us do analytical work, specifically targeted analysis, where we go out and we look for a compound in the environment, whether it be in water or sediment or even human serum or urine, asking the question, for example, this pesticide fipronil, how much of it is in my water? And CIL is obviously very well known for making not only isotope-labeled, but also native standards to develop these targeted methods. And these are best done with low-resolution mass specs like triple quads to just go and take a highly robust validated method and look for these compounds, much like shooting an arrow at a target. But higher resolution mass spectrometry really defines how we do this non-targeted analysis. And this is asking a different question. It's more like casting a net. What chemicals can we find present in the environment? And compounds can be found with instruments like QToFs and Orbitraps. And it's broken into two groups, one of which is the suspect screening, where you have a database somewhere that you can ping against your data set. You can go even after the fact, the previous presenter talked about, and mine for things that you didn't know at the time as those databases grow. But you can also do non-targeted analysis, which is what I'm going to talk about, the discovery of things that are not yet known that can then be added to a database for others to screen against and eventually be made into robust methods. And I want to set the tone and say, the first step in all the work that we do as health effect officials and people that are looking for these compounds in the environment is much like the foundation that holds your home. If you don't know what the compound is, we can't make decisions about such things as risk and occurrence and remediation. So we're sort of environmental forensic researchers trying to figure out what compounds are out there so we can then make these decisions. And the key tool that I use is high-resolution mass spectrometry. It's obviously a tool, but it's not the only tool. And when I look at a data set, I kind of think of it like a lineup, police lineup. What are the pieces of evidence I'm using to build a case for why a compound is interesting or should be looked at further? A big peak, a peak that's in many of the samples, whether or not it has additional halogens on it. I'm going to talk about this negative mass defect space. And also, I go to a lot of industrial sites and look for what is being, and I take a closer look at that data. And this is done in a series of ways. And most of the time it's done looking at the compounds upstream and downstream of a facility, like a pipe going into a river, for example, at an industrial facility, or it could even be a non-point source like an agricultural field or a golf course. It doesn't really matter how it's done. It's really by difference. And the high-resolution mass spec data can be mined to see what changes as you go down past the facility. And many of the legacy PFAS that we all know of, like PFAS and PFOA, are indicators of a reason to take a closer look. I like to always try to make the point that not everything in your data set is important, and sometimes it's okay to throw out data. And not everybody agrees with me on this, but this is a sample set where we had some effluent, and I could discover 420 or so compounds in that waste stream. And if I rank these just on a log normal scale, and I only look at the top 100 features, a lot of environmental data is log normally distributed. If we just simply start to look at what those features are and how they rank as part of the total, just that one compound alone, the number one is trifluoroacetic acid. It represents 6.5% of the total area of all those features. And as I move on down, I can identify what each of these analytes are, and there's a running total. And by the time I get to the 100th feature, so this one compound at the 100th feature, I can explain 85% of the area from those 421 compounds. So you have to ask the question, when is enough enough? And I have to make a judgment call. One of the ways we make that judgment call is looking for compounds specifically that look like they may be polyfluorinated. And so we use this mass defect. The previous speaker told you about, proteins and peptides. Carbon, hydrogen, and nitrogen are some of the elements that make those up. Obviously, there's oxygen in there. And as each of these elements is added to the organic structure, it affects the defect of the intact compound. So things like polyfluorinated species have, of course, lots of carbon, but they have many fluorines on them. And that fluorine imparts a negative mass defect. All that really means is it's slightly less than a whole number. Oxygen, phosphorus, sulfur, chlorine, bromine additionally do as well. I want to point out that one fluorine alone is really not that exciting, but polyfluorinated compounds, because they have many fluorines, are able to give you this negative mass defect. And an endogenous compound like octanoic acid, it's critical fatty acid, is absolutely something that you would find, which has a positive mass defect in the body. PFOA, the fluorinated analog, would be highly fluorinated. It has a negative mass defect. So I show there in red if it has a negative mass defect. And so we live in this continuum as analysts in the environment of trying to figure out anywhere from what's known to what's unknown, this so-called iceberg, what's underwater, or these bubble plots where we show these thousands of features. And we're somewhere in between the unacceptable and the unachievable. And I think Yogi Berra put it best: if you don't know where you're going, it's hard to kind of find out what you're up to. You need to predefine what it is you're going to do with your study. You need to predefine when enough is enough. So a little bit about PFAS. I think they live in a different realm. And I went to their ComTox database and I just downloaded some of our lists of compounds. If you don't know this database or this site, please go to this web link. I did this one, which is at the very top, it's the REACH list. And I plotted the mass defect, just basically calculating from the monoisotopic mass. And what you can see is most of these compounds, that distribution is to the right of the zero value. So the bulk of these are in the positive mass defect space. Sustat, which is another large list, is similarly to the right of this defect space. So that normal distribution differs from the PFAS list. So I want you to note that clearly the PFAS list has a different distribution. You can do a statistical test, but I think anybody could look at that and see PFAS are different. So as we go through PFAS list, this one from NIST, you can again see they differ from the bulk of the other compounds found in the REACH or the Sustat list. And if I mine down a little further, what I want to look at here is if I take that PFAS list we have, and I take away any other PFAS that has another halogen, like a chlorine, a bromine, or an iodine, this is where that normal distribution lives. If you add on the chlorine, it moves a little bit further to negative. If you add on the bromine, even further. And if you add on the iodine, it goes even further. So that negative mass defect is imparted by those additional halogens. And we again use that as a tool to try to figure out what it is to spend our time on. In this REACH database, I want to show you in the hard cut between 0.1 positive and 0.1 negative, roughly 50% of the data lives in that range. And doesn't mean that the compounds that don't live in that range are unimportant. They're very important. But from a PFAS person, looking at that space is exactly where I want to live. If we take our PFAS master list, now in that same mass defect space, we jump up to about 86% of the total data. So I want to make the point that hard cutting, and specifically to look at those features that are PFAS or appear to be PFAS, but they don't have to be, is where I want to spend my time when I distill through my data. And UCMR5 is the unregulated contaminant monitoring rule coming up soon. The 31 compounds on that list all fall within the space I've defined. And this EPA method for non-drinking water, it's either 1633 or 8327, I don't recall, also fall within that list of mass defect space. The next thing I want to point out is that industrial sites, it's okay for us to take a site monitoring well and just cut down data, where here we have 780 features that we can see. And if we only look at those with a negative mass defect space, we cut that down to about a third. If we only look at the features that are more than 5 times our blank sample, we cut that even further in half from what we saw. And we can look at those things that we have figured out before to distill down the ones that we don't yet know, to spend time on those that we do know. So cutting and eliminating is part of the job of deciding what is and is not important in some of these industrial sites. And we get a lot of samples coming in from these industrial sites where we're trying to figure out what it is that's important. We have to eliminate the what's unimportant, and that may include what's already known. Another tool we use is these in-source fragments. Many of you that have worked on polyfluorinated compounds know that they fragment at these unique locations where you can have a CF3, C2F5, C3F7, for example. You can extract up the extracted ion chromatogram for each of these. We can look for telltale perfluoroether linkages that also we can pull out from an EIC, and we can stack those up and we can see what compounds show these telltale perfluorinated fragments that we don't yet know about, and then spend time on discovering what those are. And then in this example, if I stack those all up together, if I point out this chromatogram, I can see some telltale fragments that point towards a perfluoroether compound we figured out around a Chemours facility and published in 2015, and the additional fragments that stand on top of it and are unique and diagnostic ions for these compounds. And it's not just that one compound. If we stack these up and we begin to look at measurement of repeating units using the caliper tool, we continue to see, like all polyfluorinated compounds, they never exist alone. They often exist in groups. And this CF2O repeating unit shows up again and again. And additionally, we can see a C2F4O2 repeating unit. And when those are stacked up, we can look at a homologous series of polyfluoroether carboxylic acids that were found in the downstream sample of a Chemours industrial effluent discharge that were not in the river above it. And some of these molecules were further investigated in the blood of people living downstream of that facility in Wilmington, North Carolina. So a little bit more on another tool I use, which is, if any of you have done GenX and a mass spec, what you find is that it easily decarboxylates. And this compound, which is called PFO2HXA, actually the M-H peak is shown there in red. We can see the telltale fragments that we have seen before. This 178 feature, which I'll point out in a moment, is a unique feature that happens from the rearrangement within the mass spectrometer as these compounds are fragmenting. But I also see what's called the diagnostic gas phase dimer of the sodium and the proton. This is often overlooked as a diagnostic ion. So when you do high-res work and you start to look at the molecular weight of these compounds and trying to figure out what they are, the mathematics of how things work together with that sodium and the proton are very telltale. And we published this in my 2015 study. But in this example, up in the upper left-hand corner, this diagnostic ion is just a rearrangement, which you would not anticipate coming from the original structure, where that fluorine migrates over as that compound is fragmented. And we are able to determine what it is and figure out what that structure was. So another example, we were working with the state of New Jersey, the New Jersey DEP, where they were interested in PFNA and the drinking water in the area. And the problem was that at that site, PFNA was not the biggest peak that we could find in many of those water samples. And so John Washington published this paper, one of my EPA colleagues in Science, where these compounds were found in soil. And my colleague James McCord at the EPA also published this paper where it was found in water. These chlorinated compounds, which were first reported as possible things to look for in the Zinnia and Wang paper in 2013, where you can see the solvate product. There's a Marcuse structure shown, polyfluoroether carboxylic acids with a chlorine on the end. And what we noted was in the spectra, we could see this compound in the middle shown here with one chlorine. We could see a fragment a little smaller than it that looks like it's probably just, losing a C2F2 or something like that. We could see a smaller molecule that was additional fragment, all showing one chlorine. But we could also see this dichloro compound. And as we did the math, it was pretty easy for us to figure it out from our previous work that if you just double that compound's molecular weight, add a proton to it, you have a gas phase dimer, that's a probative ion. And what you would see from that is that obviously a compound with one chlorine that dimerizes in the gas phase through a proton or a sodium would lead to an artifact on the mass spec source of a compound with 2 chlorines. It's really pointing back to the M-H peak. And as it stands, with most polyfluorinated compounds, like I said before, they don't exist alone. And so this Marcouche structure represents the propyl and the ethyl functional groups, the propyl and ethyl ether functional groups. And we could see this homologous series that showed up in the water downstream of Solvay or in their waste stream or in the monitoring wells in the area. A couple more slides about, when is mass spec, high resolution mass spec not good enough? And so we often think, well, if you've got a high res mass spec, there's no ways you can, make a determination of a compound incorrectly. And in this example, Nadine Kotlars at NC State was looking at compounds in the serum of people drinking the water from Wilmington, North Carolina. And we noted by extracting the ion chromatogram of the top compound on the top, the 6,2-FTS, and the compound on the bottom, hydroEve, one of which on the top is an AFFF formulation compound, and the one on the bottom comes from the creation of Nafion at the Chemours facility. They're within 5 ppm difference of each other. So chromatographically, just looking at that molecular weight, you can't tell them apart until you look at the isotope cluster, the sulfur 34 peak there, where we'll easily be able to distinguish that one has a sulfur and one does not. So simply having the high resolution mass spec data is not good enough. So we probed a little bit. We did some MSMS studies, which at the time we were not doing because we did not have standards for many of these. And like all of you who do 6,2-FTS work, you see the loss of an HF, the 427 to the 407. You see the loss of a sulfonate if that compound is present. But we did not see that. Our fragmentation clearly showed that it was this fluoroether hydroEve. So in this example, simply doing an EIC extract of the high-res data after the fact, you could do it post-analysis, is not good enough. The MSMS data is required in order to distinguish between the two if you can't separate them chromatographically. And here's one example that I think will show artifacts when you do non-targeted analysis. In this compound on the upper left, PFPRA, this is known as C3. And I know CIL now says this is I think it's a 3-labeled C13 compound that you can purchase for the quantitation of that PFPRA. But in this industrial monitoring well, we have a very large peak for C3. We had a very large peak for this PFMOAA. And I want to call out this M to Z 342.9. That's an unknown. So when you look at this data set, it looks like a normally distributed chromatographic peak. And as it turns out, it's not real. What's happening is we have some of the PFPRA being shown as the 162.9 peak. We see some of the PFMOAA. They're co-eluting. And in the middle, again, these gas phase dimers that we see are coming through a proton and a sodium, 2 of which we can explain because they're 2 PFPRAs coming together. And the other one we can explain because it's PFMOA coming together. But you also get this dimer in the gas phase of the PFPRA and the PFMOAA. And so this is an artifact specifically of these 2 compounds competing for ionization in the source. And I will tell you that this also happens with native compounds and isotope-labeled compounds, where you're digging through these massive amounts of data and trying to figure out what it is. And the algorithms for these pieces of software that try to figure out what that formula is, to try to figure out what that structure is, doesn't know what to do with that. So it still requires the chemists to sit there and use their brain and try to get to the bottom of what it is. So with that being said, I think there's a whole lot of tools that I've explained and mentioned in the work that I've done. There's a lot more I could go into, but I think as time allows, and I don't really think I'm right on time, I'm going to jump off and just give you my final slide. Ask me a question or contact me via email after the fact.
Thank you, Mark, for that excellent talk. Really fascinating stuff on background, really nice overview of your PFAS evaluations using HRM. We have time for 1, maybe 2 questions. If there's none from the audience, I will ask one. I'm curious what measures you currently institute to help minimize background contamination from lab materials during sample prep and LCMS sample processing, because these materials are ubiquitous of trace levels. I'm wondering if the vials, solvent lines are pre-treated, or what you do to minimize absorption.
Yeah, how long do I have? That should give you half an hour answer. And I think like everyone else, I think blank contamination is a problem. It doesn't matter who you are. All of us who have been doing this, and I know there's some speakers after me that could chime in because they do this. Andrew Patterson, for example. You have to run a lot of blanks. You have to run solvent blanks. You have to run process blanks. You have to run your instrumentation and look at what a non-inject blank looks like. That's run your gradient and don't actually inject the sample. And everything is relative to that nothing. And unfortunately, there's never nothing when it comes to PFAS in your sample. It's always a minimization. We stick with and stay with things that have been shown to work well for us: solid phase cartridges, solvents, HPLC vials, caps, etc. And if anyone changes any of that on me in the purchasing department, I get very angry because all of a sudden I have a new analyte I didn't see before as a blank, but it's just continued vigilance and just keeping on top of what is zero.
Right. Thank you for that excellent answer. With as we're right up against the time of the next speaker, we are going to move on.